I've been mostly involved in treating patients with convalescent plasma, which is the liquid portion of blood that's very rich in antibodies. So it is blood that's donated by people who've recovered from COVID-19. And then the plasma and the antibodies in it can be transfused into somebody who is actually sick at that moment with COVID, in hopes that this passive transfer of antibodies will help the sick person get over the disease faster. And so what the assay allowed us to do is to begin treating people very early in the epidemic in New York City with plasma that we knew was high titer for anti-SARS-CoV-2 antibodies. The assay was actually implemented in the clinical lab before we even had donors who had seroconverted, because you can imagine we were identifying the first positive case in New York State was on March 1st. And then that person has to recover for several weeks before their antibody titer is at its peak. And so we started screening potential donors, people that we knew had recovered from COVID, in mid-March, looking for people who had high titers. And that allowed us to refer those people to donate their blood, to donate their plasma, in order to be able to treat sick people with it. And we sort of knew that we were providing antibody-rich plasma to our